This presentation was brought to you by the Portland Psychedelic Society. Hello, my name is Eisen. I'm a graduate student at the National University of Natural Medicine, pursuing a Master's of Science in Integrative Medicine research. Integrative medicine is the combination of both naturopathic and holistic medicine that has been used for thousands of years with allopathic or Western medicine. My primary focus in integrative medicine is integrating psychedelic psychotherapy into our modern healthcare system. I'm also the education chair for the Portland Psychedelic Society, and my primary focus is promoting research, education, and community outreach within the Portland Psychedelic Society. I'm a graduate teaching assistant at Oregon Health and Science University. Primarily, I work as a statistical consultant for students in the School of Nursing who are working on their thesis projects. I'm also a research assistant at Oregon Health and Science University in a variety of different labs, including the School of Nursing, the Developmental Brain Imaging Lab, the ADHD Research Study, the CDC National Spina Bifida Patient Registry, and the Portland Alcohol Research Center. I am also the Director of Communications for Northwest Noggin, which is a neuroscience outreach group that travels to K-12 public schools and other underserved areas of our community, including Native American tribal lands, homeless youth shelters, and correctional facilities. Our primary goal with Northwest Noggin is bringing free area educational resources to underserved members of our community, providing free access to science for everyone. I am also a student member of the Society for Neuroscience, which is the largest neuroscience organization in the world. But that's enough about me. Let's get on to our main topic. We're going to have to go over a few disclaimers, so let me prepare my disclaimer voice. The views and opinions expressed in this presentation are those of the authors and do not necessarily reflect the official policy or position of OHSU, PSU, NUNM, Northwest Noggin, Society for Neuroscience, the Imperial College of London, MAPS, and MAPS Canada. Your brain is made of cells. These cells are called neurons, and they specialize in communication. As we can see by observing the structure of a neuron, it has a lot of branch-like projections called dendrites. This allows a neuron to branch out and connect with other adjacent neurons. We can also see that neurons have this long wire-like component. This is called the axon, and it allows neurons to stretch and reach out to very short or very far lengths of our brain and body. In fact, one of the longest axons that we have in our body connects from the bottom of our neck all the way down to the tip of our toes, and that is a single cell. So how do neurons communicate? Originally, there were two lines of thought. There was the chemical supers and the sparkers. The sparkers believed that neurons communicated solely through electricity, and the supers believed that they communicated solely through chemicals, which is why the supers refer to chemical soup. These chemicals are known as neurotransmitters. Modern research has shown that both lines of thought are correct. Neurons use neurotransmitters to create electricity, which then sends the signal down to the next neuron. Neurons do not fully connect to each other. There is a small gap between two neurons that are quote-unquote connecting. This gap is called a synapse, and it's where the neurotransmitters and the receptors are housed. This is also the primary site of most drug interactions. In one of my first lectures, a very, very good question was brought up to me. A person asked, so neurons communicate with one another, but what do they say? Generally, neurons have one of two things to say. They either say, turn on, and let's keep sending the message to the next neuron in the chain. This is called excitatory. There is also a message that neurons send that tell them to stop sending the message along the chain. This is called inhibitory. Essentially, neurons that are communicating tell the next neuron in the chain to either continue sending the message or stop sending the message, depending on the neurotransmitters and the type of neuron it is. As mentioned before, neurotransmitters do not connect physically when they are communicating with one another. The axon terminal of one neuron and the dendrite of another neuron form a small gap known as the synapse. The synapse is where both the neurotransmitters and the protein receptors are housed. All hallucinogens seem to act on one type of serotonin receptor, while many hallucinogens act on more than just this one type of serotonin receptor. It seems a defining characteristic of all hallucinogens is its ability to bind to this specific serotonin receptor. This receptor is known as the 5-HT2A receptor. 5-HT stands for serotonin. 2A is one of the 15 different serotonin receptor subtypes. Specifically, it is a postsynaptic receptor, meaning that it is on the dendrite of another neuron. Research has shown that the potency of a drug and its ability to bind to the 5-HT2A receptor are highly correlated. Here on this graph, we can see the potency of a drug is correlated with its affinity to bind to the 5-HT2A receptor. Down here, we can see that drugs have a higher affinity and they have a high potency. We can see that LSD has an extremely high affinity or ability to bind to the 5-HT2A receptor. This is why it's one of the most potent hallucinogens. When I was first looking at this chart, I found it was kind of odd that DMT was not as able to bind to the 5-HT2A receptor as LSD. And when you think about DMT, you think of it as being one of the holy grails of psychedelics. However, if you think about the actual dosage that you consume when you compare DMT to LSD, this relationship becomes a little bit more apparent. You take micrograms of LSD while you take milligrams of DMT. An average dose of DMT is around 100 milligrams. If you took 100 milligrams of LSD, the two trips would likely be very, very different, with LSD being much more powerful. Hallucinogens also affect the gating of sensory input. There is a very unique part of the brain called the thalamus, and the thalamus sits in a very good position right on top of the brainstem. Messages come up through the brainstem and reach the thalamus, which then sends out the messages to other regions of the cortex. I like to think of the thalamus as the old school telephone operators that would connect people's messages to one another. Here on this figure, we can see all the projections of the thalamus, starting from the central node inside of the brain and spreading out to other regions of the cortex. It seems that under the influence of psychedelics, the thalamus gets shut down. And when this operating network is shut down, we see a flurry of new communications occur in the brain. Areas of the brain start to connect that did not connect before. We start to see this local disintegration and a global integration. In these figures, we can see, not under the influence of any psychedelic, the different brain regions or lobes, which are outlined by these different colors, are communicating with each other, but not that much. Now, when the thalamus is shut down, the communication network that is kind of regulating all of these regions of the brain communicating one another, we can see that there's a flurry of new communications that occur. This may explain why psychedelics produce phenomena such as synesthesia, the ability to see sound or taste colors. We can see that different regions of the brain that might be responsible for auditory processing are now connecting to the visual regions. Or we can see that visual regions that are originally supposed to intake our vision are now connecting to our taste regions. Hallucinogens also enhance the sensory response in the locus ceruleus. The locus ceruleus is our orientation network. If you were to see a small spider running across the floor, it would be your locus ceruleus that orients you towards the spider in order to get away. Or if you stepped out into the middle of the road and a car was coming towards you rather quickly, that would orient you towards the car in order to get out of the way in time. So psychedelics tend to hijack the locus ceruleus and make you oriented to things you might not normally be oriented towards. Suddenly, ordinary stimuli become extraordinary, such as your hand, or a carpet, a pattern on a carpet, or this chair over here. So where are these receptors? Well, they're all over your brain. They're in the neocortex, which is kind of the outer region of the brain, this outer bark. And specifically, they are in layer 5. They are also found heavily in the olfactory cortex, which is smell, the hippocampus, which is memory, the basal ganglia, which can be loosely generalized as our reward network, the thalamus, which is, as I described earlier, the cerebellum, which is this region back here, which is responsible for balance, emotional regulation, and a whole host of other things. An interesting thing about the cerebellum is actually about 69% of our neurons in our entire brain are actually in the cerebellum, this small region back here. There are so many neurons in there that they actually refer to the cerebellum as the second brain. There are also high concentrations of the 5-HT2A receptor in the brainstem, as well as the spinal cord. There's also the default mode network, and the default mode network is critical for self-reflection, self-awareness, and rumination. It is the network that is working when you are wondering what you should eat for dinner tonight, or what you should do on your day off next Tuesday. You could think of it as your daydreaming network. Now we see a decreased activity of the default mode network under the influence of hallucinogens, and this promotes mindfulness and in-the-moment thinking. It may also explain why it's a little difficult to plan what you want to do next Tuesday in the middle of an LSD trip. Interestingly enough, psychedelics are not the only way to reduce activity of the default mode network. Research has shown that meditation may also reduce activity of the default mode network, which may explain why meditation is also associated with mindfulness. There are several different types of psychedelics. These would include the classical psychedelics, the pathogens and entactogens, the dissociatives, the deliriants, and the infamous research chemicals. Just want to say this is a small trigger warning. I will be showing images of a variety of different substances. So if this is something that concerns you, this might be a good time to skip ahead. Although I will say this might not be the best presentation to watch if you're not okay with being able to look at substances. First, we have the classical psychedelics, which are the psychedelics we generally think about when somebody uses that term, or hallucinogens. We have LSD, psilocybin cubensis mushrooms. There's also mescaline, ibogaine, and DMT. All of these substances are 5-HT2A receptor agonists, meaning they are known to bind to the 5-HT2A receptor and activate it. You also have the pathogens and entactogens, which include MDMA, MDA, MDE, and the whole rest of the alphabet soup. While these are also agonists of the 5-HT2A receptor, they are also reuptake inhibitors for 5-HT, which is a similar mechanism of action of SSRIs, which are selective serotonin reuptake inhibitors. So essentially, these act as SSRIs. They are also norepinephrine reuptake inhibitors, otherwise known as SNRIs, serotonin and norepinephrine reuptake inhibitors. And they are also dopamine reuptake inhibitors, which, interestingly enough, is the same mechanism of action as cocaine. They are also known to promote an oxytocin release, which may explain why these substances promote warm feelings of openness to a person's self and others. There are also the dissociatives, which include PCP, ketamine, DXM, which is a common component found in many over-the-counter cough medicines, including corticosteroid cough and cold and Robitussin, and salvia. Dissociatives are characterized for their feelings of disassociating one from their body. This seems to have a primary impact on the parietal lobe. The parietal lobe is responsible for somatosensation, or the spatial mapping of one's body. This network is what allows us to be able to touch our nose even when our eyes are closed. People who have migraines in the parietal lobe also report feelings of growing very, very large or shrinking very, very small. There was a very interesting experiment done on a woman who had her brain open and exposed in a laboratory. There are no pain receptors inside of a brain, so in theory and in practice, you can open a person's brain and perform experiments while they are still conscious. The researchers sent a small electrical stimulation to a specific network in the parietal lobe. When this network was activated through the electricity, the woman described feeling as if she was being lifted from her body, and she could actually look down and physically see her body below her. As soon as the researchers took away the electrical current, she had the feeling that she was sucked back into her body. The dissociatives also seem to act on NMDA receptors, and they tend to block activity of the NMDA receptor. The NMDA receptor is critical for both learning and memory. An experiment done a few years back with a mouse made a very, very interesting discovery about the NMDA receptor. It seems that it is open for longer periods of time when we are younger. This allows people to learn quicker and faster when they are younger, and they need to be absorbing information like a sponge. They took a mouse, an adult mouse, and they gave it a young mouse's NMDA receptors that were open for longer periods of time. With the adult mouse with the younger NMDA receptors that are open for longer, it was better able to navigate and remember mazes. Interestingly enough, dissociatives also seem to act on opioid receptors by activating them and turning them on, which may explain why PCP and ketamine and these substances are associated with a reduction of pain. Interestingly enough, PCP was originally developed as an anesthetic for surgery, but for obvious reasons, this was no longer thought viable because people were complaining of very intense psychological effects. Researchers then created ketamine to be another anesthetic, and it is commonly used in veterinarian practices because animals can't complain about psychological effects. Interestingly enough, ketamine is also used during childbirth currently in our modern healthcare system, and this makes sense because that is probably a time that somebody does not want to feel like they are in their body. We also have the Deliriants, which include nutmeg, Benadryl, Dramamine, deadly nightshade, and Datura. While most Deliriants are completely legal, these are some of the most dangerous of all the categories of psychedelics. Deliriants are both anticholinergic and antihistamines. Anticholinergic refers to the acetylcholine receptor, which is the receptor that is found at the neuromuscular junction, where neurons connect to muscle tissues. This includes the heart and lungs. At excessively high doses, deliriants can interrupt the functioning of these muscles, which can lead to cardiac arrest and other severe complications. There are also the research chemicals, which include the DOX series, the NB series, the 2C series, and a variety of other chemicals created by Alexander Shulgin. Research chemicals are incredibly dangerous as they have been recently created and have a very limited history of human use. We do not understand all the possible short-term and long-term risks of consuming these chemicals, nor do we understand the possible interactions they may have with other substances. Unfortunately, research chemicals are often legal and very easy and cheap to obtain. This has led to, unfortunately, a lot of research chemicals being sold as other psychedelics, such as 2,5-I being sold as LSD. This stresses the importance of testing your substances before you consume them. As we can see in this picture here, there's a blotter tab of 2,5-I, which looks very similar to what one might expect LSD to look like. You might not be able to read that very small label, but it says this is 2,5-I for plant food only. And that's generally how it's sold online, not necessarily as plant food, but as something that's not meant for human consumption, and then humans consume it. Although I do very much question what kind of plants they're trying to grow with 2,5-I. If you're interested in learning more about these and other substances, I highly recommend checking out Erowid, which is a drug encyclopedia. They have information on all of the substances that I mentioned in here, as well as hundreds of more substances. In further detail, the 5-HT2A receptor is one of the main excitatory receptor subtypes among the GPCRs for serotonin. In other words, the 5-HT2A receptor tells other neurons to turn on. It's an excitatory receptor. And it is also a GPCR. There are two different types of receptors. There's ionotropic and GPCR, which is also known as metabotropic. Ionotropic receptors work when a neurotransmitter binds to it. It then opens a small channel that ions pass through into the neuron. GPCRs work a little bit differently. Instead of opening and having ions go through, when a neurotransmitter binds to them, a small piece of the receptor breaks off, which is called a G protein, and then goes inside of the cell and causes a cascade of other intercellular interactions, which lead to a desired effect. Most drugs affect GPCRs, which kind of suggest these more long-term impacts of substance use. Although the 5-HT2A receptor is an excitatory receptor subtype, it seems to have inhibitory effects on certain areas, including the visual cortex and the orbitofrontal cortex. This is interesting because an inhibitory effect means that there is a lack of messages or information going to these regions. This may actually explain why psychedelics produce hallucinations. When there's a lack of information, such as to the visual network, the brain does not like this, and it fills in the missing gaps with pieces of information from other neurons. Filling in these missing gaps is what produces visual hallucinations. This also occurs in the phenomena of phantom limbs, where somebody who has lost a limb may experience that the limb is still on their body and they're still able to move it. And in some cases, people feel as if they are still in pain. There was one very unique scenario where a motorcyclist lost his arm and he was gripping onto the bike handlebars during the accident. And after he lost his arm, he had the sensation that his fingernails were still digging into his palm and he couldn't open that. It seems that the phantom limbs work in the same way that I just described visual hallucinations work. Certain areas of the motor cortex that are responsible for moving that limb connect to other regions of the brain. It may be that they connect to other regions, such as the face. So then whenever somebody moves their face, they have the perception that their arm is moving. Interestingly, this seems to be psychological, as the case that I mentioned earlier with the biker was able to be resolved not with drugs, but by a very simple experiment with a mirror. Researchers put a small mirror underneath his hand in a small little box. The mirror was connected to another researcher who had his arm. The subject put his stub arm inside of the box, and the mirror made him perceive that the researcher's arm was his own. The researcher then unclenched his fist, and the subject reported feeling as if his own fist, which was no longer there, was unclenched. The 5-HT2A receptor is highly concentrated in high levels of the cortex, which are responsible for learning and memory retrieval. It is also highly concentrated in this postingular cingulate cortex, which is a central node of the default mode network. It is also scarcely concentrated in the primary motor cortex, which may explain why psychedelics have such a strong mental impact, but barely affect movement. Now, this may be a surprise, but the 5-HT2A receptor is not solely there for psychedelic use. It has a natural purpose inside of our brain, and it is part of our stress response system. The 5-HT2A receptor naturally engages when we are in times of great stress. This promotes enhanced cognitive flexibility, improved associative learning, and hippocampal neurogenesis, or the growth of new neurons inside the hippocampus, which is responsible for memory. Activating this system helps us to overcome challenges that are in our environment and makes us be adaptable and be able to make quick, in-the-moment decisions to promote our ability to survive. The subjective effects of the 5-HT2A receptor include well-being, optimism, openness, and ego dissolution, which is very helpful when there's a need for flexibility to break rigid patterns or strongly held beliefs. Research has shown that psychedelic use may be associated with personality traits. A research study that psychedelics was associated with increased openness and liberalism. This was not found with cocaine or alcohol. The study also found that psychedelics was associated with an increased nature relatedness, which is defined as I always think about how my actions affect the environment. They found that alcohol use was actually associated with a decrease in nature relatedness. Now, when I think about liberalism, eco-friendliness, and nature relatedness, I tend to think of Portland. Now, I'm not saying psychedelics made Portland the way it is, but maybe all of these factors make Portland a very attractive hub for anybody who's interested or consumes psychedelics. Hendricks et al. In 2014 did a very monumental study that looked at recidivism and psychedelic use. Recidivism is defined as a convicted criminal who went to jail, getting convicted again for another crime and going back to jail. The results showed that psychedelic use was a greater protector against recidivism than stable family, housing, and employment. Now let's go on to the topic we've all been waiting for, microdosing. I do want to make a small disclaimer about the following sections. The literature around microdosing is rather mixed, and there are far more anecdotal reports than there are scientific research studies. The following information will only be using the scientific research studies that are around microdosing, and it will not be using the anecdotal reports unless I clearly express that the piece of information that I'm presenting is from anecdotal reports. So what is microdosing? In the past few years, the issue of microdosing psychedelics has been openly discussed in the public arena, where claims have been made about their positive effect on mood state and cognitive processes such as concentration. However, there are very few scientific studies that have specifically addressed this issue, and there is no agreed scientific consensus on what microdosing really is. The term microdosing is not a uniquely psychedelic term. In pharmacology, microdosing is a process used in drug development and drug selection, where a very small dose of a substance is used to assess the pharmacokinetics of a drug. Pharmacokinetics is the branch of pharmacology concerned with the movement of drugs in their body, or how drugs reach their receptor targets. Microdosing psychedelics has been described in a similar manner by different individuals. Fadiman describes it as a practice to use subthreshold doses of psychedelic drugs in an attempt to enhance cognitive tasks, to boost physical energy levels, to promote emotional balance, and to treat anxiety, depression, and addiction, resulting in typically subtle, though noticeable effects. Similarly, Alet Waldman in her book states the same intention for microdosing, but describes the process as the act of integrating sub-perceptual doses of psychedelic drugs in your weekly routine. In addition, Johnstad emphasizes that to microdose with the psychedelic drugs means to take a small dose enough to provide no intoxication or significant alteration of consciousness. It seems that the term microdosing appears to consist of three primary components: the use of a low dose below the perceptual threshold that does not impair normal functioning of an individual, a procedure that includes multiple dosing sessions, and the intention to improve the well-being and enhance cognitive and/or emotional processes. A microdose has been defined as approximately 1/10 to 1/20th of a recreational dose, varying within and between substances, so it can be seen as somewhat being a very low dose. Although microdosing of psychedelics does not have an agreed upon scientific definition, we have decided to continue to use the term because of its prevalent societal use. People who microdose mainly follow one of three approaches. The most popular of these is the Fadiman approach, outlined in his book, which involves 2 consecutive dosing days followed by 2 non-dosing days. Another popular approach involves weekday dosing, for example, from Monday to Friday, but not dosing on Saturday and Sunday. Additionally, some users indicate that they follow a balanced low microdose approach, which involves dosing every other day. Dosing periods often range from 1 week to 2 years. This variation in microdosing schedules was confirmed by a recent survey, which demonstrated that half of respondents who microdose came up with their own dosing schedule and did not use any of the other 3 common approaches. So what dosing protocol is right for you? Unfortunately, as with most psychopharmaceuticals, there is no standard dosing regimen that works for everyone. This is true for many common psychopharmaceuticals such as SSRIs. No single dosing regimen or dosing amount works equally well for everyone. That is why we have psychiatrists, so people can attend meetings with psychiatrists who evaluate their current status and help them to determine the appropriate dosing schedule and amount for their symptoms. In other words, the only way to know what works for you is through personal experimentation. However, it's important to note there's a large variety of factors that may influence how well a microdosing protocol works for you. These may include, but are not limited to, the potency of the psychedelic, your diet depending on the psychedelic, weight, height, genetics, liver enzymes such as cytochrome P450, mental health status, personality type, and what's called natural tolerance, which we'll talk about a little later on. Research has shown that common microdoses for a variety of different substances are as follows. Psilocybin is generally less than 1 milligram. LSD is generally somewhere between 10 to 25 micrograms, but 25 micrograms may produce some subjective effects. Ibogaine is around 200 milligrams, and DMT is around 6 milligrams. However, it's important to note the dosing regimen of a microdose protocol varies not only by which substance you use, but also the potency of the substance. 2 people who consume the very same substance, such as LSD, may have very different doses if the potency of the LSD is different. For example, if somebody had an LSD tab that was 50 micrograms and another person had one that was 200 micrograms, each of those people would take a much different size piece of the tab to take a microdose. The most widely distributed species of psychedelic mushrooms are key cubinescents, and those of the genus Coplandia, which consist of 12 species. It is very important to note that each genus and species of mushroom varies in the amount of psilocybin it contains. Furthermore, the potency of each species and genus can vary depending on a variety of factors, such as the time of its collection, where it was grown, the soil quality, how it was dried, and how it was stored. User-reported recreational doses depend on the species and the experience of the user. A hallucinogenic dose of dried cuminescens, for example, is generally between 3 and 5 grams. These values equate to a recreational dosing range of 8.6 to 14.7 milligrams of psilocybin per dose. Thus, a microdose would range from 0.43 to 0.73 milligrams of psilocybin per dose, because a microdose of psilocybin is generally 1/10 of a full dose. That positions a recreational dose of psilocybin between a low and medium dose and a microdose below a very low dose. However, variations in psilocybin content between doses of dried mushrooms may be seen due to variations between individual fungi within a species. LSD is a semi-synthetic psychedelic substance that commonly comes in the form of a blotter tab, a liquid, gel tabs, a pill, or a microdot. While LSD does not have a variety of different genuses and species like mushrooms do, it comes with its own set of challenges. First off, LSD is notoriously dangerous to handle, and I say this more in reference to the liquid form than any of the other forms. A vial of LSD can be dangerous to handle, as even a single drop of it on your skin can cause profound psychoactive effects that may be unexpected or unwanted. Moreover, accidents do happen, and spilling an entire vial of LSD on yourself would be similar to taking a dose that far exceeds a normal recreational dose. LSD is also very difficult to handle. It degrades in the presence of light, heat, and oxygen. Therefore, we have to store LSD away from these elements. Improper storing techniques can cause LSD to degrade much, much quicker. LSD is also extremely potent, which can make it difficult to microdose with. My best recommendation for this is for liquid LSD. I recommend putting a single drop of liquid LSD in a water bottle and slowly drinking it over the course of a few days, or whatever complies with your microdosing regimen. We also have to be concerned, as I mentioned before, with the presence of fake LSD. As I mentioned, many research chemicals such as 2,5-I are commonly come in the form of blotter tabs, and as they are very easy to acquire and sometimes legal, and they are cheap to obtain, there are people out there, unfortunately, who are selling them as LSD. That stresses the importance of using testing kits to make sure you are actually microdosing with the substance you think you are. As I mentioned before, there are also variations in potency. One tab of LSD might have 100 micrograms, while another might have 200 micrograms. We can see that the size of the chunk of the tab that somebody would take would be very different between these two tabs. You might be wondering, what is more beneficial, dosing microdosing LSD or microdosing psilocybin? Unfortunately, to my knowledge, there is only one study to date that has looked at this question. The study was done by Anderson et al. In 2019, and it compared outcomes between psilocybin-only microdosers and LSD-only microdosers. The results showed that the psilocybin-only microdosers reported the benefits as significantly more important than the LSD-only microdosers. Furthermore, the psilocybin microdosers did not report any more challenges than the LSD microdosers. Tolerance is a natural phenomenon that occurs with substance use, as in microdosing regimens. Tolerance can be either natural or it can be acquired. There's very little research on natural tolerance to psychedelics, but this phenomenon has been anecdotally reported in many online forums. Current research points to the theory that variations in our serotonin network, which contains the 5-HT2A receptor, may explain why some people are more sensitive to the effects of psychedelics compared to others, even at the same doses and potency. Another theory presented by Dr. Groff suggests that the degree of sensitivity or resistance to LSD seems to depend on complicated psychological factors rather than on variables of constitutional, biological, or metabolic nature. Dr. Groff states that subjects who in everyday life manifest a strong need to maintain full self-control and have difficulties in relaxing and letting go can sometimes resist relatively high doses of LSD in the range of 300 to 500 micrograms and show no detectable changes. This may suggest that the personality trait of openness may play a role in natural tolerances to psychedelics. Occasionally, individuals can resist considerable doses of LSD if they have this set as a personal task for themselves. Additional causes of high resistance to the effects of the drug may be insufficient preparation, instruction and reassurance of the subjects, a lack of their full agreement and cooperation, or an absence of basic trust in the therapeutic relationship. Instant sobering, which can occur at any period of a psychedelic session and on any dosage level, typically indicates a sudden mobilization of defenses against the impeding emergence of unpleasant traumatic material. Among psychiatric patients, severe obsessive-compulsive neurotics are particularly resistant to the effects of LSD, although that is rather controversial, and I have seen studies that have both found that to be true and found that not to be true. It has been a common observation in Dr. Groff's research that such patients can frequently resist doses of more than 500 micrograms of LSD and only show slight signs of physical or psychological distress. This may suggest that the mental health status is related to natural tolerance to psychedelics. It is important to note that this theory is oriented towards psychedelic psychotherapy, but the basic principles apply to microdosing as well. Diet, weight, liver enzymes, and height may play a role in both natural and acquired tolerances, but more so for psychedelics that are orally ingested, such as mushrooms and ibogaine, rather than psychedelics that are taken through other methods of administration, such as LSD through the buccal method, which is generally between gum cheeks or under your tongue, and DMT that is smoked. The repeated administration of psychedelics, such as in microdosing regimens, leads to a very rapid development of tolerance known as tachyphylaxis, which is a type of acquired tolerance. This is a phenomenon believed to result from 5-HT2A receptor downregulation. The figure on the right describes receptor downregulation. Up at the top of the figure, we can see that we have stars which represent agonists, which would be neurotransmitters, or in our case, a psychedelic substance. We can see that the psychedelic substance then binds to different receptors, such as the 5-HT2A receptor. Now remember, the 5-HT2A receptor is a GPCR receptor, or a metabotropic receptor, which means that when an agonist, like a psychedelic drug, binds to it, it then releases G proteins that go inside of the cell and cause intercellular changes. These intercellular changes result in less gene expression of receptor proteins and cause there to be less 5-HT2A receptors in the synapse of a neuron. We can see this represented in the bottom figure here, where there are much less 5-HT2A receptors than before all the agonism occurred. Research has shown that daily administration of LSD leads to essentially the complete loss of sensitivity to the effects of the drug by day 4, when the dose is kept the same every day. 5-HT2A downregulation may also explain why cross-tolerance occurs. As all psychedelic drugs, such as LSD and psilocybin, bind to the 5-HT2A receptor, any one of these substances can cause downregulation to occur. When downregulation occurs, you have less receptors for the psychedelic to bind to, which causes there to be less effects. In other words, if somebody continuously dosed LSD, they would downregulate their 5-HT2A receptor. And then if they consumed another psychedelic, such as psilocybin, there would be less receptors for psilocybin to bind to, meaning they would have effectively created a cross-tolerance for psilocybin from their LSD use. Using the basic theory of receptor downregulation, PsyCal created a tryptamine redosing calculator to provide an estimated dose to achieve the same effects of taking a tryptamine while considering acquired tolerance. However, the results of this calculator should be taken with a grain of salt, and I do not necessarily condone its use. It assumes that a person's tolerance will be back to baseline after 14 days, and it does not consider any of the other factors that I mentioned in the past slides. In summary of the past few slides, it's your responsibility to educate yourself, to research the safety, the efficacy, the dosage, the contraindications, and any side effects of microdosing regimens. Everyone responds differently to psychedelics, so just because one dosing protocol works for someone else does not mean it will work for you. You will have to figure out on your own what substance, what dose, and what dosing protocol works best for you. That hopefully will help to prevent this from happening, where you start feeling effects where they were not wanted or not expected. It's important to note that if you are microdosing, you are leaving the seemingly protected and precise world of big pharma. You will have to figure out the safety of microdosing on your own by doing extensive research. You'll also have to find a reliable source. For example, dried mushrooms are safer than pills, and at least you'll know you're eating mushrooms. You will also have to figure out your dosing. We recommend starting at 0.1 grams of psilocybe cubanescens if you are the anxious type, and 0.2 grams of psilocybe cubanescens if you are the deep end of the pool kind of person. We can provide you with recommendations, and the microdosing group is a valuable resource to share information and discuss questions that you might have. But in the end, you shouldn't trust us either. There is simply not enough research on the topic for us to provide you with a valid answer. The pharmacology of psilocybin is still unclear due to the rapid decline of psychedelic drug research following them being made Schedule I drugs in the 1960s. This decline predated the growth of modern neuropharmacology. Thus, more research is required to build a more complete pharmacological profile of psychedelics and microdosing. To get more information, we recommend the following resources. This would include Erowid, as I mentioned before, YouTube, theshroomery.org, and a whole host of other websites. You can also contact me if you want me to send you a research article that was discussed in this paper, or if you have any other questions. My email is provided in the slide below. So now let's talk a little bit about the effects, the drawbacks, and safety of microdosing. In a recent study, researchers looked at the psychological effects of microdosing psilocybin. There were 10 outcome categories in this study, and each outcome category had its own corresponding benefit and challenge. I thought this was a very unique and interesting way to approach the research question. In this figure, we can see the prevalence of the different microdosing benefits compared to the microdosing challenges within the research study population. In the main analysis, the researchers compared the differences in the main outcome categories between the prevalence of the benefits and challenges associated with that outcome category. As we can see here, positive values indicate greater endorsement of benefits over the challenges. So this line represents 0, and we can see that all of these variables reported more benefits than challenges, while these variables over here reported more challenges than benefits. The researchers also looked at behavioral improvements and substance use behavioral reductions associated with microdosing psilocybin. As we can see here, the purple upward-facing triangle represents improved behaviors, versus the downwards pointing orange triangle represents reduced consumption of a substance. We can see that alcohol, caffeine, cannabis, illicit drug use, psychiatric drug use, and tobacco all decreased with microdosing regimens. We can also see that anxiety improved, eating behaviors improved, exercising behaviors improved, meditation improved, mood improved, and sleep quality improved as well. A recent literature review, which summarized all of the available literature to date on microdosing, described the psychological effects of microdosing that have been regularly reported by users after multiple administrations of psychedelics. The users reported improvements in energy, mood, cognition, concentration, management of stress, creativity, spiritual awareness, productivity, language capabilities, relationship, and visual capabilities. The users also reported reductions in anxiety, depression, addiction, and pain relief. Other research has also shown that psychedelics are associated with low-level associative learning, extinction learning or unlearning, and the flexibility of behavior and thought patterns. However, these three characteristics were found in macrodosing trials, not microdosing trials. However, the characteristics of these three variables likely pertain to microdosing as well. Research has also shown that psychedelics can produce beneficial behavioral effects without drastically altering one's perception. Research has also shown that microdosing psychedelics has a variety of physiological effects. These effects include increased neuroplasticity in the cortex, or the growth of new neurons. Interestingly enough, research has shown that microdosing promotes neuroplasticity more than larger doses of psychedelics. Microdosing also promotes synaptogenesis, which is the growth of the functional components of neurons, such as the axons, dendrites, and axon terminals, which we can see outlined in the figure to the right. Same with neuroplasticity, it seems that microdosing promotes synaptogenesis more than with larger doses of psychedelics. Microdosing is also associated with anti-inflammatory effects in peripheral tissues and slight changes in heart rate. A study in 2013 by Catlow et al. Explored the relationship between the extinction of fear conditioning and the dose-response curve of psilocybin. The researchers used the properties of stimulus conditioning to train the rats to fear a small auditory cue. The researchers placed the mice in a small cage and observed their movements for 3 minutes. The researchers then played a small tone and sent a small shock to the floor of the cage 30 seconds after the tone was played. This was repeated 10 times each day for 3 days. At the end of the 3 days, the mice would freeze out of fear whenever they heard the tone, regardless of whether or not the shock was administered. This suggests that the fear conditioning was successful. The researchers then separated the mice into 5 groups: a control group that took no psilocybin, a very low dose group, a low dose group, a moderate dose group, and a high dose group. The results showed that the rats injected with low doses of psilocybin, or the microdosing group, extinguished cued fear conditioning significantly more rapidly than the high-dose psilocybin or control group mice. Specifically, rats in the high-dose and control groups no longer froze to the tone by trial 10 with no shock. However, the rats in the very low-dose group, or the microdose group, no longer froze to the tone by trial 3. The results of this were replicated by Cameron et al. In 2019. However, DMT was used in place of psilocybin in that study, yet the results were still the same. A recent study in 2018 explored the subjective psychological effects of a high dose of psilocybin compared to a low dose of psilocybin. The researchers looked at convergent thinking, which is persistence over flexibility, divergent thinking, which is flexibility over persistence, and creativity. In the high-dose group, the researchers noted an improvement in divergent thinking and creativity, and a decrease in convergent thinking. However, in the low-dose group, researchers noticed an increase in divergent thinking, convergent thinking, and creativity. Research has shown that the 5-HT2A receptor can have an indirect modulation over dopamine networks. What this means is when a psychedelic binds to the 5-HT2A receptor and activates it, it can cause cellular changes that impact dopamine networks. This is true even though the 5-HT2A receptor is a serotonin receptor, and dopamine networks use the dopamine neurotransmitter. One common misconception about dopamine is that it's the reward neurotransmitter. This is not entirely accurate. Dopamine is actually the motivation neurotransmitter that plays a critical role in movement. If we were hungry and there was a pizza sitting on the counter, it would be dopamine that is motivating us to stand up and go walk over to the counter. In another scenario, if somebody were drowning in a body of water and somebody threw them a life raft, it would be dopamine that is motivating them to swim over to the life raft to save their life. For us coffee drinkers out there, for those of you who wake up early in the morning and you're not fully awake and you just kind of hover into the kitchen and get the coffee pot going without thinking and you're kind of on autopilot, that's dopamine motivating you to move, to set it up. Now, dopamine playing a critical role in movement, also plays a critical role in the development of several pathologies, such as Parkinson's disease. In Parkinson's disease, one of the dopamine networks called the nigrostriatal pathway is damaged. Specifically, a structure in the nigrostriatal pathway called the substantia nigra. When the substantia nigra is damaged, it prevents dopamine from being able to flow through that network, which results in hand tremors and other finite movement issues that we see in Parkinson's disease. A common treatment for Parkinson's disease is a drug called L-DOPA, which is actually a precursor for dopamine. By prescribing people L-DOPA, the brain has more of the raw ingredients to create dopamine, and thus there is more dopamine in the nigrostriatal pathway, which seems to reduce the symptoms of Parkinson's. It's important to note, though, that with L-DOPA medication, there's lots of warning labels, including one that says, do not go out gambling while you are under the influence of L-DOPA, because you'll have a lot more dopamine in your system. Interestingly enough, a lot of drugs that release dopamine, such as amphetamines, cocaine, MDMA, other substances like that, they seem to be stimulants. And it seems that dopamine is actually converted, or it's a precursor to norepinephrine and adrenaline. So it seems that increasing dopamine also increases our adrenaline and our ability to get up and move. So that's why dopamine plays a very critical role in motivation and movement. There are also a lot of common misconceptions about serotonin as well. Serotonin is believed to be responsible for mood, although it's much, much more complicated than that. The actual term serotonin responds to blood thinner, and actually 90% of serotonin is found in your gut. Now, while serotonin does play a role in mood and other things similar to that, it seems that it's not that simple of a relationship. Increasing serotonin does not necessarily mean you will have increased positive mood. It seems that it's very dependent on the context of one's environment. Research has shown that people in more enriched environments, when they increase serotonin, receive more positive effects from that environment. However, people in more negative environments, when they have increased serotonin, tend to experience more negative effects of that environment. This may explain why SSRIs, which their primary mechanism of action is increasing serotonin, might not work well for everyone. We have to consider who is able to access and get treatment, probably the people in the more enriched environments. As I digress, the 5-HT2A receptor for microdosing also is associated with enhanced motivation and focus, and reduced distractibility, procrastination, and rigid thought patterns or behaviors. To the right, I created this graph, which kind of describes the relationship and the modulation between 5-HT2A activation and dopamine. Specifically, this graph looks at how dopamine levels relate to performance and function. And we can see that the graph is in an inverted U-shaped curve, meaning that at low doses, there's low levels of performance and function, versus at excessively high doses, there is also low levels of performance and function. This may explain why drugs such as methamphetamine, which release a lot of dopamine, do increase performance and function, but as a person uses them for extended periods of time, they tend to not be able to perform or function that well. It seems that there's this kind of middle ground of dopamine, this kind of Goldilocks principle of not too much or too little, promotes the ultimate performance and function capabilities of our brain. And this is what we see in microdosing, which may explain why people reportedly in the Silicon Valley are using microdosing to get over writing blocks and code blocks that they deal with in their daily work. It might also suggest that microdosing may have specific benefits for depression treatments as well. However, the high dose has its own benefits as well, as it tends to cause kind of this cognitive reset to occur, which may play a role in addiction treatment and other things like that. Although it's important to note that microdosing may have benefits for addiction as well, and high doses may also have benefits for depression treatments. This is part of the nature of psychedelic psychotherapy. Not a single Just as microdosing regimens don't work for everyone, a single dosing regimen of a psychedelic may be appropriate for some people with some conditions versus people with other conditions. There are several drawbacks also reported by users who microdose. These drawbacks include the illegality of psychedelics, the stigma associated around psychedelics, physical discomfort associated with taking psychedelics, anxiety, which may be related to the actual anxiety of the substance or maybe anxiety around taking the substance, overstimulation, cognitive interference, emotional difficulty, and the uncertainty of the effect, or not knowing if something really happened or not. Consuming psychedelics, including microdosing regimens, does come with its own set of dangers and risks, although these risks are oftenly overreported by the media, such as in the examples we can see on the screen. My personal favorite is "Girl gives birth to a frog, and doctors blame LSD." Compared to most substances, psychedelics are rather safe to consume, regarding the therapeutic threshold and addiction potential of the substances. And this is compared to many other substances that are common in our society, including caffeine, alcohol, and nicotine. As with any substances, there are risks for microdosing psychedelics. With proper technique, education, and a degree of caution, these risks can be drastically reduced. And generally speaking, a small dose is associated with less negative effects. The safety risks of microdosing come more from the effects of long-term use rather than a single dosing session. This is why it's so important to find a dosing protocol that actually works for you. To discuss what the therapeutic threshold is, I will have to describe a few different terms. First off, drugs have two classes of effects. This would include the therapeutic effect, or the beneficial effects, otherwise known as the reason why somebody is taking a drug. And then there are the side effects, which are all the unwanted effects or non-therapeutic effects of a substance. It's important to note that these effects are interchangeable, depending on the reason why someone consumes a drug. For example, opioids are a class of drug with known effects such as pain relief and constipation. If somebody was in pain and took an opioid, the therapeutic effect would be pain relief, and the side effect would be constipation. If someone had diarrhea and took an opioid, the therapeutic effect would actually be the constipation, but the side effect would be pain relief. In this example, we can see how the same substance can have different therapeutic and side effects depending on why somebody is consuming the drug. The therapeutic effect refers to the dose of a substance that is required to feel some kind of beneficial or therapeutic effects. The toxic effect is the dose of a drug that is known to produce toxic effects in at least 50% of the population tested. The therapeutic index is the difference between the therapeutic effect and the toxic effect. We want this difference to be larger, as a larger difference would describe a safer drug. For example, if we had two drugs, drug A and drug B, and drug A had a therapeutic effect that was noted at 1 milligram and a toxic effect that was noted at 1,000 milligrams, versus if we had drug B, where a therapeutic effect was also noted at 1 milligram, but a toxic effect was noted at 3 milligrams. Obviously, drug A is much safer to consume. This figure describes the safety of several psychedelic compounds compared to a variety of other substances commonly consumed in our society. We can see that on the x-axis, the active dose versus lethal dose is reported. This is otherwise known as the therapeutic index. As I described in my previous slide, we want the difference between the active dose and the lethal dose to be larger, which would suggest that it's a safer drug. It's important to note that the active versus lethal dose in this figure is reported as a ratio. Given that it's expressed as a ratio, we want this ratio to be smaller, which would suggest a safer drug. Based on the x-axis, we can see that substances on the left half of the screen are safer than the substances on the right half of the screen. The y-axis describes the dependence potential or addiction potential of all these different substances. Higher up on the figure describes a greater addiction potential versus lower on the figure describes a lower addiction potential. Based on this, we can see that LSD and psilocybin have a much lower addiction potential and are even safer to consume than other commonly consumed substances, including caffeine, nicotine, and alcohol. In one of the lectures I attended, I heard one of the researchers who was presenting said that LSD was safer than drinking water, which is an interesting line of thought. His justification for that was that you can take 1,000 doses of LSD and not die, but you can't drink 1,000 glasses of water because you would die. While this kind of generalizes it a little bit, it's a very interesting perspective to consider. A dose-response curve is a measure of the relationship between the dose of a drug and the response or effect that that dose produces. A threshold is defined as a dose that produces the first measurable response or the first noted effect of a drug. The ED50 describes the dose that produces half of the maximal effect of a drug. As we can see, drugs have a maximal effect where their effect tends to plateau even with increasing doses. When the maximal effect is reached, even increasing the doses will not increase the effects that drug has on a person. Drug potency is defined as the amount of a drug or a dose needed to produce a response or an effect. Drugs that have larger effects with smaller doses are more potent. As we can see in the figure, drug 2 is more potent than drug 1, and this is because we see that the ED50 and the maximal effects are reached at a smaller dose than drug 1. Drug efficacy is described as the effectiveness of a drug in producing the desired response. Drugs with a higher maximal effect are more efficacious. As we can see in the figure, drug 1 is more efficacious than drug 2, as it has a higher maximal effect than drug 1. And this is true even though drug 1 requires a larger dose than drug 2. It is still more efficacious than drug 2. In line with media reports and anecdotes, the majority of people who microdose tend to do it to enhance performance. Negative effects that occur are mostly acute and happen directly after substance administration. However, the main reason that people tend to stop microdosing is not due to negative side effects that occur, but rather that the microdosing regimen was not effective. The most pressing dangers of psychedelic use, beyond the legality of it, is that psychedelics can cause profound cognitive and behavioral changes. These cognitive and behavioral changes can cause people to engage in dangerous activities that can and have led to fatalities. Psychedelics can also cause profound life-changing experiences that may be unexpected or unwanted. However, these dangers are acute in nature and do not pertain to the low doses associated with microdosing. As mentioned before, the primary concerns with microdosing is the effects of repeated use, which are not acute in nature. Research has shown that common complaints around microdosing are related to both physical and psychological components. The physical components include headaches, nausea, dizziness, and tiredness, and psychological complaints generally include paranoia, anxiety, and depression. To date, there has never been any serious adverse reactions in any microdosing study. However, this does not mean that a serious adverse reaction is not possible. It just means that we don't have any evidence of them happening. Obviously, more research is needed on the safety of microdosing. Preclinical studies to assess the safety of repeated doses of psilocybin in rodents have not been thoroughly conducted. That may be due to several factors, including the historical background of psilocybin as an ingredient in magic mushrooms that has been used by many cultures without apparent harm. Evidence from these accounts demonstrates a lack of serious adverse events resulting from psilocybin administration. While there has never been any serious adverse event reported in any psychedelic microdosing study, research suggests that a serious adverse event is possible. An adverse cardiovascular event due to an HERG potassium channel blockade from psilocybin is possible, but the chances of that happening are extremely low at microdosing levels. An HERG analysis demonstrated minimal effects of psilocybin at concentrations up to 1,000 micrometers and completely without effects at 100 micrometers. This means that unwanted cardiac chronotypic effects with microdosing is very unlikely, as the maximum plasma concentration of psilocybin produced by a 25 milligram dose would not even reach 160 micrometers. Another potential serious adverse event are cardiac valvopathies due to repeated activation of the serotonin 5-HT2B receptor, which psilocybin activates along with many other serotonin receptors. It remains to be seen whether repeated low dose of psilocybin administration in preclinical studies might produce valvopathies, and whether or not this would translate to the human user population. So far, psilocybin testing in preclinical studies has not revealed any signs of valvopathy. However, even with repeated microdosing, there is a possibility that 5-HT2B receptors might be stimulated enough to lead to tissue overgrowth. LSD is a common psychedelic that is often used for microdosing purposes. It has been examined in rodent models after repeated dosing schedules that are similar to microdosing protocols. Comparatively, low doses of LSD administration every other day for several months were shown to produce persistent negative behavioral changes that lasted for at least several weeks to months after the LSD administration was discontinued. These changes include increased aggression, scruffy appearance, the inability to feel pleasure, and hyperreactivity. An analysis of gene expression in key cortical regions like the medial prefrontal cortex indicated that LSD produced alterations in genes enriched for schizophrenia and bipolar depression that lasted long after the drug was discontinued. Of note, the interspecies scaling questions arise, and it is disputable whether the low dose used in animal models are comparable to those used by humans. As I mentioned before, psychedelics tend to act on the 5-HT2A receptor, but they also act on a variety of other receptors, including the 5-H2B receptor and the 5-HT1A receptor. Therefore, some stimulation at the 5-HT1A receptor may occur during microdosing regimens. Such activity has been implicated in the mechanism of action for antidepressant medications, including SSRIs, which suggests that there could be interactions between psychedelics and SSRIs, MAIOs, or other medications that impact serotonin system. Due to the lack of research, we do not understand all the possible drug interactions between microdosing psychedelics and taking other pharmaceuticals. One possible interaction that may occur is known as serotonin syndrome, due to a dramatic increase in serotonin in the central nervous system, which could potentially lead to death. In summary, microdosing psychedelics is relatively safe, and there has never been any reported serious adverse reactions to my knowledge. While there are some risks, the chances of a serious adverse reaction is very low. These risks can be properly managed with proper techniques, education, and a degree of caution. As everyone responds to psychedelics differently, you will have to find what works for you through your own self-experimentation. A good rule of thumb is to always start with less and work your way up until you feel comfortable. Keep yourself educated on current literature that comes out and share resources with other fellow microdosers. Sometime in the near future, the Portland Psychedelic Society will start collecting data on microdosing trends within our community in order to perform a study that may help us to answer some of the many questions we have about microdosing. If you are interested in participating in the study, please stay tuned. In the meantime, happy explorations. Thank you.